Hello students, welcome to EPG Pathshala. I am Fauzia Zakir from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Hamdard. Today we are going to discuss about module Nasal Delivery of Polar Drugs from paper Novel Drug Delivery System 1. So students, let us see what we are going to learn in this module. We will first start with nasal mucosa permeability and the factors affecting drug absorption. We will then pass on to challenges in nasal permeation. We will also study why morphine can be administered by intranasal route with the help of a case study. Polar drugs are water soluble and charged molecules, but they are lipid insoluble. As we know, like dissolves like, non-polar drugs as compared to polar drugs gain access across lipid membrane. Nasal mucosa is made up of lipid membrane which is selectively permeable in nature. Hydrophobic and small polar drugs can diffuse through nasal mucosa, but ions and large polar drugs can diffuse to very less extent. Lipid bilayer contains integral membrane protein through which ions and large polar molecules can pass through by active or passive transport. Membrane protein transport hydrophilic drugs protein channel is often gated to regulate the transport rate. Facilitated transport, which is a type of passive diffusion, allows hydrophilic molecules to pass through carrier protein. Hydrophilic molecules also pass through active transport and uses the carrier protein, but there is a requirement of energy as diffusion is against the concentration gradient. The figure in front of you describes the movement of drugs across the lipid membrane. The molecules can diffuse across a membrane down a concentration gradient without the aid of a protein or input of energy. Passive and active transport requires membrane transport proteins. Channel proteins carry out passive transport, whereas carrier proteins can carry out passive or active transport. The factors which affect the absorption of drug through nasal mucosa are ionization, lipophilicity, surface charge, hydrophilicity, molecular weight, and size of the molecule. Ionization. pKa of drug and pH of nasal mucosa determines the degree of ionization. Drug should be in unionized form for its permeation across nasal mucosa. So drug should be buffered so that nasal mucosa pH does not produce any change in formulation pH. Lipophilicity. Nasal permeation is directly proportional to log P of drug. Greater the partition coefficient of drug, greater will be the log P value. Drug should be lipophilic so that it can easily cross the nasal mucosa. Surface charge. Some polymers have inherent surface charge like that of chitosan, which has positive charge on its surface. But other drug delivery polymers for which surfactants are used as stabilizers shows the charge imparted by surfactants only. Surfactants used should be such that it provides positive charge on the surface of polymer because mucin is negatively charged. So therefore, the positively charged formulation can adhere to the nasal mucosa electrostatically, thus increasing the residence time of formulation in nasal cavity. Hydrophilicity. Hydrophilic drugs are poor candidate for permeation through nasal mucosa. The permeation can be modified chemically by the use of prodrugs. A prodrug of ester or amide, depending upon the free functional group available for modification, can be used. Prodrug being lipophilic can be then available for absorption. Another way is to coat the drug by lipophilic polymer like PLGA, or the drug can be delivered in the form of certain formulations such as liposome, nanoemulsion, or solid lipid nanoparticles. molecular weight. The molecular weight of the drug should be less than 1000 Dalton for its easy absorption. High molecular weight proteins and peptides like insulin and calcitonin are not a good candidate for absorption. Size. Small sized drugs in nano range such as nanoemulsion, liposome, solid lipid nanoparticles are easily transferred through nose to brain because the trigeminal and olfactory neuron diameter is around 200 nanometer and the particle size lesser than this can be easily transferred to the brain. The added advantage is that these nanoformulations are made of lipid which can easily cross nasal mucosal barrier. Low molecular weight hydrophilic drugs are absorbed through the nasal mucosa at the rate comparable to intravenous route. High molecular weight, where the molecular weight is more than 1000 Daltons, hydrophilic drugs are not easily absorbed through nasal mucosa. Proteins and peptides like insulin, calcitonin, and leuprolide, which are high molecular weight hydrophilic drugs, have bioavailability less than 1%. Apart from the membrane permeability and mucociliary clearance, these proteins and peptides also suffer from proteolytic enzyme degradation present in nasal mucosa. Modification of the chemical structure of the molecule to make it non-polar, which is also known as prodrug approach. Prodrug approach is mainly meant for optimizing favorable physicochemical properties such as solubility, taste, odor, stability, etc. Prodrug is usually referred as pro-moiety as it is to cover the undesired functional group with another functional group. This prodrug approach is mainly used for improving the nasal bioavailability, especially for the proteins and peptides. This approach enhances the membrane permeability along with increased enzymatic stability. The prodrug undergoes enzymatic transformation to release the active medicament when it crosses the enzymatic and membrane barrier. The absorption of peptides like angiotensin II, bradykinin, choline, carnosine, enkephalin, vasopressin, and calcitonin are improved by preparation into enamine derivatives. These agents show improved absorption with prodrug approach. Modification of drug structure without altering pharmacological activity is one of the lucrative ways to improve nasal absorption. The chemical modification of drug molecule has been commonly used to modify the physicochemical properties of a drug such as molecular size, molecular weight, pKa, and solubility. These are favorable to improve the nasal absorption of drug. For example, chemical modification of salmon calcitonin to icatonin showed better bioavailability than salmon calcitonin. This is because the CN bond in the salmon calcitonin has been replaced by the SS bond to produce icatonin. Prevention of enzymatic degradation by using suitable enzyme inhibitors along with drug in the formulation is another approach to improve nasal permeation. Use of permeation enhancers like surfactants, bile salts, cyclodextrins, chelators, and bioadhesive materials to increase the penetration of polar molecules is also an important parameter to improve the permeation of high molecular weight proteins and peptides. Permeation enhancers are mainly used for the enhancement of absorption of the active medicament. Generally, the absorption enhancers act via one of the following mechanisms. They inhibit enzyme activity, reduce mucus viscosity or elasticity, decrease mucociliary clearance, open the tight junctions present in the nasal epithelium. They also solubilize or stabilize the drug. The mechanism of action of absorption enhancer is increasing the rate at which drug passes through the nasal mucosa. Many enhancers act by altering the structure of epithelial cells in some way, but they should also accomplish this while causing no damage or permanent damage to nasal mucosa. Drug delivery system is also another alternative. Particle design is an increasingly important role in absorption enhancement. Microspheres, nanoparticles, and liposomes are all systems which can be used as carriers to encapsulate an active drug. The properties of this can be varied to maximize therapeutic efficacy. Overall, this can result in increased absorption efficacy and stability and reduced toxicity of the active ingredient. Systems can be designed to be mucoadhesive to increase the retention time and facilitate sustained release. Microspheres are mainly Microspheres mainly increase the absorption and bioavailability by adhering to the nasal mucosa and increase the nasal residence time of the drug. The microspheres prepared by using polymers like dextrin, chitosan, biodegradable starch microspheres successfully improved the bioavailability of various drugs. Liposomes are amphiphilic in nature and are well characterized for favorable permeation of drugs through the biological membranes. So the water-soluble drugs, which are difficult to permeate, have been delivered by liposomal delivery to the nasal mucosa. Cationic liposomes are having good permeation capacity than negatively charged anionic liposomes. Use of P-glycoprotein inhibitors like poloxamer 188, sorbitol HS15, cremophore RH40 and polyethylene glycol also improve the permeation of drug through the nasal mucosa. We will now discuss the various approaches to overcome the challenges in nasal permeation. Modification of the chemical structure of the molecule to make it nonpolar, which is also known as the prodrug approach. Prevention of enzymatic degradation by using suitable enzyme inhibitors along with drug in the formulation. Use of permeation enhancers like surfactants, bile salts, cyclodextrins, chelators, and bioadhesive materials to increase the penetration of polar molecules. Another strategy is entrapping the molecule in suitable carrier, which is known as novel drug delivery system. Use of P-glycoprotein inhibitors like poloxamer 188, sorbitol HS15, cremophore RH40, and polyethylene glycol. Chitosan is a copolymer of glucosamine and N-acetyl glucosamine. Amine groups of chitosan is protonated upon dissolution, so the chitosan is cationic in nature. Chitosan is available in the broad range of molecular weights and salt forms such as glutamate, lactate, and chloride. The physiological properties of the chitosan, for example pKa and solubility, can be modified by changing the degree of deacetylation, pH, and ionic strength. Chitosan is positively charged polysaccharide and is bioadhesive due to the amino group which form bond with negatively charged mucus secretion of nose, which is mainly sialic acid. Modification of the chitosan like trimethylated, pegylated, carboxylated, and thiolated chitosan can change and improve the properties of the chitosan such as its charge, charge density, solubility at higher pH, and change in pKa value. For example, Calu-3 cell monolayer showed reduced toxicity with M-polyethylene glycol coated chitosan as compared to unmodified chitosan. Chitosan has improved transmucosal delivery of many challenging drugs, including low molecular weight polar drugs with marked improvement in their bioavailability, such as morphine and anti-migraine drugs, as well as peptides such as calcitonin, desmopressin, goserelin, parathyroid hormone releasing hormone, and leuprolide. Chitosan can be used for delivery of liquid and powder formulation as well. Drugs which are not stable in solution, such as parathyroid, can be delivered by entrapping in chitosan. Chitosan, being hydrophilic in nature, is used to deliver hydrophilic molecules like selegiline, zidovudine, propanirole, etc. Presence of intercellular tight junctions makes hydrophilic drugs impermeable to cell of size larger than gap junction and which cannot diffuse through lipid bilayer. Mechanism of bioavailability enhancement of these drugs using chitosan as carrier is due to bioadhesion of chitosan to nasal mucosa and transient opening of tight junctions of cellular epithelium in nasal mucosa. Chitosan affects the ion transport channel by interacting with the cell surface. Such interaction cause the activation of intracellular events like involvement of second messenger and protein kinase that regulates the opening of tight junction. Chitosan, due to its positive charge, is able to interact with the opening mechanism of the tight junctions as seen by decrease in zona occludens 1 proteins and change in the cytoskeleton protein F actin from a filamentous to a globulal structure. Mucoadhesive nature of chitosan has been evaluated by gamma scintigraphy of chitosan, which can increase the half-life of clearance of liquid and powder formulations from 15 to 45 and 85 minutes respectively through nasal mucosa. Chitosan glutamate solution was used for increasing the transport of insulin across nasal mucosa in sheep. It was found that the level of glucose in plasma decreased to 43% in 90 minutes with 0.5% concentration of chitosan glutamate. There was 7.2 times increase in the AUC of intranasal administered chitosan insulin formulation as compared to insulin solution. In another study, chitosan nanoparticles were compared with chitosan solution and simple powder. It was found that as compared to solution, chitosan nanoparticle showed about 3 times lesser bioavailability while powder formulation had 5 times higher bioavailability. This result was due to nasal clearance half-life of approximately 115 minutes for powder formulation when compared to 43 minutes for solution. Goserelin was administered intranasally to sheep using 3 different formulations, which are chitosan solution, chitosan powder, and lyophilized chitosan microsphere. In another study, goserelin was administered intranasally to sheep using 3 different formulations, which are chitosan solution, chitosan powder, and lyophilized chitosan microsphere. Bioavailability of these formulation as compared to subcutaneous injection was found to be 11.8%, 25.6%, and 36.6% respectively. Different absorption enhancers were compared with that of 1% chitosan solution in rat for nasal delivery of thiamopentin. Bioavailability of 46% was obtained with chitosan solution without any toxicity to nasal mucosa as compared to 5% bridge and 5% deoxycholate. It was also observed that absorption did not increase when enzyme inhibitor bacitracin in a concentration of 0.5% was used. Chitosan alnidatin formulation was intranasally administered for the treatment of migraine and a bioavailability of 60% was observed in phase 1 trial. Later, in phase 2 studies, 2 doses of formulation were administered during and outside migraine attacks, and it was observed that early rise in plasma concentration of alnidatin related directly to headache improvement and that the measured pharmacokinetic parameters were similar during and outside migraine attacks. This study shows that the formulation is well tolerated in the patients. Morphine can be used through intranasal route. We will now discuss the rationales for using morphine. Morphine is an alkaloid. It is used in case of pain and is a narcotic which is obtained from opium. Oral absorption of morphine is very low, which is 1/6 to 1/4 of the parenterally administered medicine. Biodistribution of morphine is high and its concentration in liver, kidney and spleen is higher than plasma. Its plasma protein binding capacity is 30%. These factors limit the fraction of morphine which is available to the brain. Being polar, morphine has additionally more limitation in penetrating the blood-brain barrier. So oral route is not as effective as the parenteral route for administering morphine due to obvious limitation of the oral route. However, morphine is a good candidate for administering it through intranasal route if compared to intravenous route. This is supported by the study which is done by Vestal, who showed that the brain to plasma ratio of morphine for 0 to 5 minute period after intranasal administration in rats was higher than intravenous administration, indicating early distribution of morphine via nasal route. Nasal mucosal barrier can resist the absorption of the morphine as the drug is polar. So the drug can be encapsulated into nanoparticulate system to overcome this limitation. The added advantage of nanoparticulate system is that it can provide sustained release profile, thus reducing the dosing frequency of morphine. One such chitosan-based nanoparticulate system of morphine has been discussed. The figure in front of you shows the pain score versus time after intranasal administration of chitosan morphine formulation to cancer patients in a pilot study for the treatment of breakthrough pain. Chitosan formulation of morphine showed 5 to 6-fold increase in bioavailability as compared to morphine solution in sheep. In human intranasal formulation of chitosan, morphine was compared with intravenous infusion of morphine for 30 minutes. A bioavailability of 60% was obtained with a Tmax of 15 minutes, indicating rapid and effective absorption of morphine. The shape of the plasma profile for nasal delivery of chitosan morphine formulation was similar to the one obtained for the slow intravenous administration of morphine. The next figure in front of you shows morphine plasma concentration in human volunteers after administration of morphine as chitosan solution and human and powder formulations through nasal route. Analgesic efficacy and safety of a novel intranasal morphine formulation, immediate release oral morphine, intravenous morphine and placebo in a post-surgical dental pain model was compared. Intranasal morphine doses were similar to the intravenous and oral morphine, and all 4 morphine treatments were statistically superior to placebo. Overall, intranasal morphine 15 mg presented an efficacy profile similar to intravenous morphine 7.5 mg. Both treatment demonstrated rapid onset of efficacy, generally persistent throughout the 6-hour assessment period. The lower dose of intranasal morphine 7.5 mg was statistically similar to the active treatments at 2 hours and 6 hours and similar to placebo at 4 hours. In other study, morphine 6 glucuronide was administered as chitosan solution in sheep, either nasally or as an intravenous injection. The bioavailability of the nasal formulation was found to be 31.4%. So far, no pharmaceutical nasal product has been marketed containing chitosan as an absorption enhancer. But one nasal morphine chitosan product, Rylomine, which is a product of Javelin Pharmaceuticals, has been through phase 3 clinical trials. Javelin Pharmaceuticals announced that its product candidate Rylomine met its primary clinical endpoint of achieving better sum of the pain intensity difference scores in the time period of 0 to 24 hours after surgery. In this first of two pivotal phase 3 studies, patients with moderate to severe pain after elective orthopedic surgery were given a 7.5 milligram dose of Rylomine through as compared to intranasal morphine every hour as needed, a 15 milligram dose every 3 hours as needed, or a 7.5 milligram intravenous morphine dose every 3 hours as needed. Patients in each of the three active treatment groups had significantly better SPID scores over 24 hours than the corresponding placebo groups, according to the company. In this first of the two pivotal phase 3 studies, the patients with moderate to severe pain after elective orthopedic surgery were given a 7.5 milligram dose of Rylomine every hour as needed, a 15 milligram dose every 3 hours as needed, or 7.5 milligram intravenous morphine dose every 3 hours as needed. Patients in each of the three active treatment groups had significantly better SPID scores over 24 hours than the corresponding placebo groups, according to the company. There were no serious adverse events related to the study drug. Local adverse events, that is bad taste, throat irritation, instillation site reaction, nasal erythema, and nasal irritation, were more common with Rylomine or intranasal placebo than the intravenous morphine groups, the company said. General systemic adverse events related to active drug were similar across morphine treatment groups, regardless of route of administration, and consistent with morphine's known pharmacological effects, for example, dizziness, nausea, transient decreased oxygen saturation, and vomiting, according to the company. It was concluded in this study that Rylomine demonstrated statistically superior analgesic activity compared to placebo and similar analgesic activity and safety as compared to parenteral morphine. Rylomine exhibited a linear dose response within the dose range appropriate for the treatment of acute moderate to severe pain. Rylomine 7.5 mg is equivalent to 5 mg intravenous morphine. Rylomine 7.5 mg and 15 mg were both effective, with the larger dose showing superior efficacy and smaller dose showing better tolerability. Safety outcomes suggest that Rylomine can cause morphine-like adverse events typically seen with other forms of parenteral morphine. Rylomine carries the risk of other forms of parenteral morphine analgesia along with their benefits. So students, let us summarize what we have learned in this module. We have first discussed polar drugs being hydrophilic, so they can gain access through nasal mucosa through passive diffusion or active transport, although the access is difficult. Low molecular weight drugs can pass easily as compared to high molecular weight drugs, various techniques to enhance permeation of polar drugs. We have also discussed the role of chitosan in nasal drug delivery. Finally, we have discussed the delivery of morphine through nasal route. Thank you.